So today I'll be talking about molecular tools in healthcare. This might be a difficult topic, especially if you're not familiar with the techniques in molecular biology. But I'll try to make this as simple as possible, and I'll integrate a lot of the topics that you should have taken during class. Now, the most important thing to remember here is that you have the central dogma, right? That's from DNA to RNA to protein. So, this is the flow of information in biology, and at each level, you can have different pathologies that will lead to clinical manifestations. Now, for DNA, you can have SNPs or single nucleotide polymorphisms. So, SNPs are single base pair mutations that can confer some pathologic activities to your DNA either by increasing its function or decreasing its function. You can also have rearrangements. So, rearrangements can be, for instance, when you have rearrangements of chromosome 9 and 22 in chronic leukemias, right? And you can also have problems with regulation. Now, problems with regulation are typically epigenetic in nature, okay? And since they're epigenetic in nature, it means that you don't really change the sequence of the DNA or rearrange your sequence of DNA. What can happen here is you can have DNA methylation. Now, DNA methylation usually leads to the silencing of the expression of DNA. If you methylate it, if you recall, right, that DNA is negatively charged because of its phosphate group, okay, PO4 3 minus. And methylation, right, this confers a positive charge to here, and so you can't transcribe your DNA. Histone acetylation, on the other hand, okay, so you can also affect the expression of DNA at the level of the histone. If you acetylate your DNA, okay, acetylation, so let's say this is your histone, okay, if you acetylate it, you're conferring negative charges to your histones. So if you're conferring negative charges to your histones, which are typically positively charged, it won't, it will set your DNA free, essentially. So your DNA, which is negatively charged, it will be free from your histone. Whereas if you methylate this, if you can imagine, if you methylate this, you're keeping, okay, the DNA in. So acetylation is activation, and methylation is repression. So you can assess these different changes in the DNA for the SNP, okay, and for rearrangement with simple PCR. I can discuss PCR in a later video, but for epigenetic changes, you can't assess that with PCR. You need to do what's called bisulfite sequencing. Bisulfite sequencing to see these changes in the epigenome. Alright, so now let's move on to RNA, okay, changes in RNA. So it's possible that you have RNA upregulation because RNA is made from DNA, and if you have upregulation, it means you're producing more of the gene. Okay? And downregulation, this obviously means that it's going down, right? So if you can upregulate the gene, what happens there is, if for instance it's a tumor suppressor gene and you upregulate it, you'll have you have better protection. If the upregulated gene is an oncogene or a proto-oncogene, it means it can be cancerous. Then you can develop certain cancers because you're upregulating an oncogene. Whereas if you downregulate a tumor suppressor gene like P53, for instance, so P53 is a well-known tumor suppressor gene. If you downregulate P53, and you won't be able to stop certain cancers from developing, then you can develop cancer. Okay, so that's the importance of knowing whether your transcript is upregulated or downregulated. How do you measure this? So, one, you can do RT-qPCR. Okay? RT-qPCR is reverse transcription quantitative PCR. So, sometimes you might hear RT-PCR, which stands for real-time PCR, but it's a bit confusing because real-time and RT and reverse transcription are both RT. However, we usually say qPCR if it's quantitative and RT for reverse transcription. Microarray studies can also be done. This typically involves the use of fluorescent markers, which I can also discuss in a later video, or you can search that up on your own. And after we go to the level of RNA, you can have the level of protein. Now, at the level of protein, you can have differences in post-translational modifications, and you can also have changes in the amino acid sequence. Now, presence or absence of protein can be confirmed with a Western blot. Okay, it can be confirmed with a Western blot. Now, just a historical note, it's called a Western blot because originally, a guy named Edwin Southern, developed a method for sequencing DNA, and molecular biologists, trying to be funny, I suppose, stuck with the directions for naming sequencing. Okay, so for RNA, we have northern blots. For proteins, we have western blots. And for eastern blots, we have post-translational modifications. But that's just a historical note. Take note that these are the names of sequencing for these different biomolecules. Now, ELISA is enzyme-linked immunosorbent assay, is a more quantitative way of determining protein expression. So this involves antibodies. Western and ELISA both involve antibodies, but ELISA is what we call a colorimetric assay, usually. Colorimetric assay. So this can be, this is assessed by looking at the change in color of a solution. So this is done usually through enzymes. Okay, enzymes that are linked to antibodies. Sometimes your ELISA can be conjugated to fluorescent molecules. And this is usually done in more advanced laboratories. So how does the principle of ELISA work? So in ELISA, you have, let's say you have your protein, and you have a primary antibody that will stick to this protein of interest. So it will stick to that protein of interest. Okay, let's draw a couple of antibodies there, right? And here, it has multiple epitopes for a secondary antibody. Okay, let's put the secondary antibody there. Okay, and these are usually linked to what we call fluorophores. So, fluorophores, or it can be linked to an enzyme, and addition of a substrate can change the color of the substrate, signifying the presence of the protein. Now, quantifying this change in color, okay, leads to the quantification of the protein. Okay, so you typically do this in a 96 well plate, right? So there's a lot of different proteins, and you can have primary antibodies sticking here to the protein, and it has multiple epitopes for a secondary antibody that's conjugated to an enzyme or a fluorophore. Now, we can also do mass spec. Okay? Mass spectrometry is analyzing the different molecular components found in a protein. You can degrade the protein amino acid by amino acid and try to see the different amino acids that's present in your protein chain. You can also have a lateral flow assay. So a lateral flow assay is essentially a strip, okay, that's conjugated with some antibodies and some gold nanoparticles, and you can assess whether a protein is present or not just by putting in some sample. So if you put in some sample, you can test positive by, for example, you have pregnancy tests. Pregnancy tests are lateral flow assays. There are monoclonal antibodies present there, and if you have the dreaded positive, right, in two lines in a pregnancy test, this is because you have certain antigens binding to the monoclonal antibodies present in your lateral flow assay, then you can have a positive result. So this is a brief overview of the molecular tools in healthcare. In the next video, I might be talking about PCR and QRT-PCR plus microarray to help understand what's going on in this rather complex field of molecular tools in healthcare.